Hi everybody, I'm Dr. Rajam Krishna, Professor of Physiology, Chettinad Hospital and Research Institute. I'm here to talk about neurotransmitters and sleep. Let me begin my lecture with the quotation from William Shakespeare, which says, "Sleep that knits up the raveled sleeve of care, the death of each day's life, so labor's bath, balm of hurt minds, great nature's second course, chief nourisher in life's feast." What does this mean? Even in the 16th century, it has been known that sleep is a very important fundamental physiological phenomenon that helps in restoring the normal vital functions of the body. So it is a homeostatic process occurring to restore the vital functions of the body. So all of us must have learned about the functions of sleep and the mechanisms inducing sleep in the initial few lectures. By now, you would have realized that sleep is not just a passive process which occurs as a result of fatigue. It actually involves a complex set of neurons which act like a switch between the awake state and the sleep state. So let's learn more about these mechanisms in this lecture as well. So now, what are the learning objectives for this lecture? What are neurotransmitters? What is their role in modulation of the sleep-wake cycle? Why should we learn about these neurotransmitters? Is it important? Does it have any clinical implications? So these are all the learning objectives of this lecture. Let's see this one by one. What are neurotransmitters? The name itself says neurotransmitters. So they are synthesized in the neuron. So they are chemicals synthesized in the neuron. And where are they synthesized? In the cell body of the neuron. They are synthesized and they are packaged in small vesicles, transported along the axon, and they are released at the nerve terminal. So at the nerve terminal, there is a junction between two neurons which is called the synapse. That is where it is released, or it is also released at the junction of a nerve and a muscle. So these are neurotransmitters, classical neurotransmitters. And the neurotransmitter, what is the purpose of this neurotransmitter? So it serves as a communication between two neurons, and this communication is a chemical communication between two neurons. Let's see about the functional anatomy of a chemical synapse. So this is a chemical synapse. So there is a presynaptic neuron and a postsynaptic neuron. So this is the presynaptic neuron and that is the presynaptic terminal. So at the presynaptic terminal, you can see the vesicles, transmitter vesicles, which are seen at the presynaptic terminal. So these contain the neurotransmitter in the form of vesicles packaged and present at the nerve terminal. And where are these vesicles released? They are released into the space between the two neurons, which is called the synaptic cleft. Following the synaptic cleft, there is the postsynaptic neuron. So on the postsynaptic neuron are present the receptor proteins, which are the receptors for the neurotransmitter. So the receptors of the neurotransmitters are present on the postsynaptic membrane. That is where they go and act. So this is the functional anatomy of the chemical synapse. Let's quickly revise the steps involved in transmission across a chemical synapse. Whenever there is an action potential in the presynaptic terminal, what happens is this action potential, when it reaches the nerve terminal, there is entry of calcium through voltage gated calcium channels. So calcium enters. So what is the role of calcium? Once calcium enters, that is the trigger for exocytosis to happen. So we know that the neurotransmitters are present in the synaptic terminal inside the vesicle, and the exocytosis of the neurotransmitters, that happens in the synaptic terminal. Exocytosis, from exocytosis, as a result of exocytosis, neurotransmitters are now released into the synaptic cleft. Once released, these neurotransmitters, where do they go and act? They go and attach to the receptor proteins which are present on the postsynaptic membrane. So these receptor proteins are nothing but channels by themselves. So they are called ligand gated. So the receptor goes and binds to these the ligand goes and binds to these receptors, and they open these channels. So if there is an entry of sodium inside or a positive ion like calcium inside, so that results in an excitatory postsynaptic potential. So there is depolarization on the postsynaptic membrane, thereby resulting in excitation on the postsynaptic membrane, which is called the excitatory postsynaptic potential. Suppose this neurotransmitter is not, the receptor protein does not allow the entry of a positive ion inside. Instead, a negative ion enters. Negative ion like chloride enters, chloride influx, or it could be because of potassium efflux. So all these are the reasons for hyperpolarization. Hyperpolarization occurs on the postsynaptic membrane. So if hyperpolarization occurs on the postsynaptic membrane, that results in an inhibitory postsynaptic potential. So if it is excitation on the postsynaptic membrane, the neurotransmitter what is released is an excitatory neurotransmitter. And if the neurotransmitter what is released, if that is going to cause inhibition on the postsynaptic membrane, that neurotransmitter is called inhibitory neurotransmitter. So this is the brief introduction to the neurotransmitters and the transmission occurring across a chemical synapse. Having studied about that, let's now look at the classification of the neurotransmitters involved in sleep and wakefulness. How are they broadly classified into? These neurotransmitters can be broadly classified into an excitatory and inhibitory neurotransmitters. We saw that in the previous slide itself. And most of these excitatory neurotransmitters are the ones which are responsible for the awake state. And the inhibitory neurotransmitters are the ones responsible for promoting sleep. And how are these further classified? Excitatory neurotransmitters are classified into small molecule neurotransmitters and a neuropeptide. So what is a neuropeptide? This has a slight difference from a classical neurotransmitter. It is not a classical neurotransmitter. So when I say classical neurotransmitter, what is it? So the classical neurotransmitters, they are packaged as small vesicles and they are released in the nerve terminal, and the mode of action is fast, and they are the ones responsible for causing potential changes on the postsynaptic membrane directly. But this is not a classical neurotransmitter, the neuropeptide. So they are also synthesized in the neuron, released at the nerve terminal. They are packaged in large dense core vesicles. The mode of action is very slow, and they do not cause direct changes on the postsynaptic membrane. Instead, they modulate the changes happening on the postsynaptic membrane. So that is how there is a slight difference between a classical neurotransmitter and these neuromodulators or neuropeptides. So how are these small molecule neurotransmitters classified further? So there is a group called the acetylcholine neurotransmitter, which comes under the acetylcholine, and then bioamines and amino acid neurotransmitters. Again, minor structural differences between bioamines and amino acid neurotransmitters. And bioamines are further classified into catecholamines, serotonin and histamine. So these are all the neurotransmitters which are classified under bioamines. Under catecholamines, you have two major groups, that is norepinephrine and dopamine neurotransmitter. Amino acid neurotransmitter, you have the glutamate, which is an amino acid neurotransmitter which is excitatory. A neuropeptide, must have heard about hypocretins. So orexin is a type of neuropeptide which forms which is classified under a neuropeptide, which actually acts as a neuromodulator. So all these are excitatory neurotransmitters. So what are all the excitatory neurotransmitters? You have acetylcholine, norepinephrine, dopamine, serotonin, histamine, glutamate and orexin. Coming to the inhibitory neurotransmitters, which are the ones responsible for promoting sleep, these are classified into amino acid neurotransmitter or a neurohormone. So this neurohormone, which I told you, this is actually synthesized in the neuron and they are not released into the synaptic terminal. Instead, they are released and taken up by the bloodstream. They act on distant sites. So the amino acid neurotransmitter, the major amino acid neurotransmitter, GABA. gamma-aminobutyric acid and galanin. So, these are the amino acid neurotransmitters, of which GABA is the major one. Just like glutamate, which is the major excitatory neurotransmitter, here GABA is the major inhibitory neurotransmitter. Under neurohormone comes the melatonin. All of you must have heard about melatonin. Melatonin of late is being studied a lot. We will be seeing more about melatonin. Then comes the neuromodulator adenosine. So, this is also an inhibitory neurotransmitter responsible for promoting sleep. So, these are all the neurotransmitters which are involved in sleep as well as in awake state. So, having studied about the classification, basic classification of these neurotransmitters, now let us see where are these neurotransmitters produced, synthesized, and which neuronal groups are they produced. So, there are neural centers which promote arousal and sleep. So, let us connect these centers to the neurotransmitters and let us study. So, the neural centers which are responsible for arousal and sleep. So, the arousal promoting centers broadly comes under the category of reticular activating system, which is the ascending reticular activating system. Similarly, you have sleep inducing areas as well. So, what are the systems which come under this ascending reticular activating system? So, you have two major groups of neurons, that is a cholinergic pathway and then the monoaminergic neurons, which come under the ascending reticular activating system. So, under the cholinergic pathway, you have the brainstem nuclear group in the upper pons. So, all these are names of the nuclear groups which you should be knowing. By the way, all of you must be knowing what do you mean by nuclei when I say nuclei. So, these are large collections of cell bodies of neurons present inside the central nervous system. So, in the upper pons, you have the pedunculopontine tegmental nucleus and the laterodorsal tegmental nucleus. So, these groups of neurons, they release the neurotransmitter acetylcholine, hence are classified under this system of neurons. Coming to the monoaminergic set of neurons, where are they present? So, if you classify the monoaminergic neurotransmitters, there are noradrenaline, serotonin, histamine and dopamine, which comes under this monoaminergic system. Where are these neurotransmitters synthesized? So, in the lower pons as well as in an area in the posterior hypothalamus. In the lower pons, locus coeruleus and dorsal and medial raphe nucleus. So, that is in the pons. They secrete the neurotransmitter noradrenaline and serotonin respectively. Coming to the posterior part of hypothalamus, the histamine secreting neurons present in the tuberomammillary nucleus. And in the midbrain, there is a nuclear group called the ventral periaqueductal gray secreting the neurotransmitter dopamine. So, all these form part of the monoaminergic system. So, now from here, where do these neurons go and how do they cause the excitation? So, you have the brainstem nuclear group in the upper pons, which usually sends the information to the thalamus and from the thalamus to the cerebral cortex. So, thalamus to the cerebral cortex is always kept under excitation. And here they go through the lateral hypothalamus again to the cerebral cortex. That is how they keep it excited and activated. So, these neurons are the ones responsible for keeping the person alert, awake. Okay, so now coming to the sleep inducing areas. Where are these sleep inducing areas situated? The sleep inducing area, this is a very important nucleus which all of you should learn and remember, which is the ventrolateral preoptic nucleus. Where is it present? In the anterior hypothalamus. This is a very important nucleus responsible for sleep induction. And there is another nuclear group in the basal forebrain, which is the median preoptic nuclei. So, what about these neuronal groups? What neurotransmitter do they release? So, they are all GABAergic neurons. All of us learnt about the neurotransmitters, and we know that GABA is what neuron? It is an inhibitory neurotransmitter, and this is the major inhibitory neurotransmitter released in these centers which are responsible for inducing sleep. And which phase of sleep are they responsible, or when do they fire? They fire maximally during non-REM sleep. So, you must have been introduced to the different phases of sleep already. So when a person moves from an awake state to a sleep state, they enter into a phase of sleep which is called non-rapid eye movement sleep. And there are different stages of this, which you will be learning more about it in the forthcoming lectures. So, and these cells fire maximally during this phase, and hence they are called non-REM on cells. These cells are active and they fire during the non-REM phase. And from a non-REM phase, every sleep cycle is interrupted by another phase of sleep which is called the REM phase of sleep. What are the neuronal groups responsible for the REM phase of sleep? In the pons, nucleus reticularis, pons oralis and caudalis, which are cholinergic neurons which secrete the neurotransmitter acetylcholine, and the sublaterodorsal nucleus in the pons secreting the neurotransmitter glutamine. Okay, yes, glutamine comes under excitatory neurotransmitter. We will see how it is responsible for inducing sleep, because these are the centers classified under sleep. So all these are REM on cells. So you have a non-REM on cell and a REM on cell. So when a person moves from an awake state, they enter into the non-REM phase, which is interrupted by the REM phase. So, and these are the REM cells which fire, REM on cells which fire maximally during REM phase of sleep. Now, what about these systems in the ascending reticular activating system, and when are they active? Yes, so they are all responsible for awake state. So they fire maximally during wakefulness. Apart from that, this you should remember, these cells in the cholinergic pathway, not only they fire during the waking or the alert stage, they also fire during another phase of sleep, which is the rapid eye movement sleep or the REM sleep. So they are also responsible for REM sleep induction. Okay, we will be seeing more about these mechanisms in the forthcoming slides. I'll tell you more about it. So now we know these are all the centers. So all these nuclear groups, yes, of course, you should be knowing about these nuclear groups which are all involved in the mechanism of arousal and sleep. So now you must be having a mental picture of these nuclear groups. Where are they located? Mainly in the brainstem and then in the hypothalamus. So now let's connect it to the picture and let's learn more about the centers. The same centers I'm going to show you here so that you will know where these centers are located. This is a schematic representation, and what you can see is the pons here and then the medulla here. And these centers are all situated here as well as in the region of the hypothalamus. So let's look at these centers. So these are the two nuclear groups. Remember, they are all what neuronal groups? Cholinergic neurons, which are in the upper pons, the pedunculopontine tegmental nucleus and the laterodorsal tegmental nucleus. And the locus coeruleus, dorsal and median raphe nucleus, which secrete the neurotransmitter noradrenaline and the serotonin. So they are all situated in the lower pons. And there is a nuclear group in the posterior part of the hypothalamus, which is the tuberomammillary nucleus secreting the neurotransmitter histamine. And the dopamine is secreted by a nuclear group in the midbrain, which is the ventral periaqueductal gray. And one more, the orexin neurotransmitter. It is a neuropeptide, neuromodulator. Where is it secreted? In the lateral hypothalamus. So all these are excitatory neurotransmitters which are seen in which part? In the hypothalamus, posterior lateral hypothalamus, and then in the upper lower pons on these nuclei and the midbrain as well. So all these are the neurotransmitters and the nuclear groups which are involved secreting the neurotransmitters. What are they responsible for? They are all responsible for awake state. That is why I have shown you in green. Coming to the centers which are responsible for inducing sleep. Yes, I've told you, remember, this is the center which you should not forget, which you should remember, which is the ventrolateral preoptic nuclei. Where is it present? In the anterior hypothalamus. And one more in the basal forebrain, median preoptic nuclei. So they secrete the inhibitory neurotransmitter GABA, and they are responsible for induction of sleep. And which phase of sleep? REM sleep. So they are all REM on cells. And coming to the that is non-REM sleep. So they are all non-REM on cells. That is the ventral preoptic nuclei and the basal forebrain median preoptic nuclei. So, they are all responsible for the non-REM phase of sleep. Coming to the REM phase of sleep, what are the nuclear groups involved in REM phase of sleep? So, in the nuclear group which extends from the caudal to the rostral pons, that is the nucleus reticularis, pontus oralis and nucleus reticularis, pontus caudalis. And remember, I told you they are all cholinergic neurons. They secrete the neurotransmitter acetylcholine. They are all REM on cells. And one more nuclear group which is present in the pons. So, that is the sublaterodorsal nucleus secreting the neurotransmitter glutamate. Both these nuclear groups are involved in REM sleep. And which phase of REM sleep? So, this nuclear group in initiation of REM sleep, and if you look at the sublaterodorsal nucleus, what are they responsible for in the REM sleep? Inhibiting the motor neurons in the spinal cord. They are responsible for decreasing the muscle tone, which is an important feature of REM sleep, and that is called REM atonia. So, REM atonia. So, this nuclear group is the one which is mainly responsible for the induction of REM atonia. So, the decreased muscle tone which occurs in REM sleep. So, now what have we learned so far? We have now learnt about the classification of neurotransmitters and where are these neurotransmitters synthesized in which neural centers, and then how are they connected. So, let us see more about the connections now in the next slide. So, in the neural circuitry of sleep and wakefulness, how are each of these centers connected and how do they work? So, we know more about these centers now. So, let us restrict now to the monoaminergic neurons which are responsible for the awake state. So, that is the locus coeruleus, raphe nucleus and the tuberomammillary nucleus, which forms part of the brainstem reticular formation responsible for the awake state. And what about the nuclear group which is involved in inducing sleep? So, from here you have the connections going to the thalamus and to the cerebral cortex, which keep the cerebral cortex active and thereby responsible for the alertness of the individual. And the sleep inducing center, especially the non-REM inducing center, where is it situated? In the anterior hypothalamus and the basal forebrain, ventrolateral preoptic nuclei and the median preoptic nuclei. So, now how are these connections? So, if you look at these connections, so this is a awake center and this is the sleep center. And how are these connections? If you look at these connections, you can see mutually inhibitory connections. So, from the ventrolateral preoptic nuclei, you have inhibition of the brainstem nuclear group, and the brainstem nuclear group inhibits the ventrolateral preoptic nuclei. So, that is how these connections are. So, now coming to the orexinergic neurons which are situated in the lateral hypothalamus. Remember, we have already seen this is a neuromodulator. So, more than their direct connections to the cerebral cortex, they have a main role in modulating the activity of the brainstem neurons. So, orexin plays a very major role in that, in modulating the activity of the brainstem neurons, and thereby they keep the excitation of the brainstem neurons, and thereby they are responsible for the alertness of the individual. And this orexinogenic neurons are kept under inhibition by this ventrolateral preoptic nuclei. So, when this is active, this is going to keep the orexinogenic neurons inhibited. So, that is how these connections work. And then we have also seen about the dopaminergic neurons which are present in the ventral periaqueductal gray of the midbrain, and through their receptors, how do they work? They directly cause excitation of the thalamocortical fibers. Not only that, they keep this ventrolateral preoptic nuclei inhibited. And these orexinogenic neurons, they not only act on the brainstem nuclear group, they also excite the dopaminergic fibers. So, that is how they modulate the activity happening in the brainstem nuclei which are responsible for sleep and wakefulness. So, this is the neural circuitry which is involved. And then, because I have already told you, these are mutually inhibitory connections. So, scientists have done a lot of research in sleep, and a lot of animal studies and mathematical modeling they have done. As a result of all their discoveries, they found these mutually inhibitory connections. How do these connections work? So they described a model or a hypothesis which is called the flip-flop switch model of sleep and wakefulness. So this is actually a concept in electrical engineering, how a switch works in electrical engineering. So there is an abrupt change of state. Similarly, here too the switch works in the same way. So there is an abrupt change of state from an awake state to a sleep state, from a sleep state to an awake state. So, and then these mutually inhibitory connections, how do they work? When there is more activity in one set of neurons, that shuts off the activity in the other set of neurons. So that is why they described this model as a flip-flop switch model. So that means when there is more activity in this, say for example, more activity in the brainstem nuclear group, that shuts off the activity in the ventrolateral preoptic nuclear group. So this is what it is called the flip-flop switch model of sleep and wakefulness. So this is a neural circuitry and the connections involved with the various neural centers and the neurotransmitters. Yes, so we have seen about these connections. Now let's see how is this switch working during an arousal state or during wakefulness. So what happens during wakefulness? During wakefulness, which are the set of neurons which should be active? So you want the neurons in the brainstem nuclear group to be active. So the activity of this brainstem nuclear group, that has to increase. So what actually promotes the activity of the wake promoting neurons? A lot of hypotheses are there. One accepted hypothesis is the activity of the suprachiasmatic nucleus. So what is this suprachiasmatic nucleus? I am introducing you to another nuclear group called the suprachiasmatic nucleus. This is also present in the hypothalamus, and this is the one which sets the circadian rhythm. What do you mean by the circadian rhythm? So all of us have a 24 hours cycle in a day. What do? We sleep for so many hours and then we remain awake for so many hours. And there is a rhythm. We have a rhythm which is set. Yes, around this time we sleep and then we wake up at this time. So this rhythm setter is the suprachiasmatic nucleus, which sets the circadian rhythm. It is also called the biological clock. And how it works, we will see that in the forthcoming slides. So this input from the suprachiasmatic nucleus, excitatory input, goes to these orexinogenic neurons in the lateral hypothalamus. What will orexinogenic neurons do? They will activate the brainstem nuclear group and then they will activate the dopaminergic nuclear group as well. So by causing excitation in the wake promoting neurons, we've already described that is a switch. How does it work? When the activity of one set of neurons increases, the activity of another set of neurons decreases. So when the activity in the brainstem nuclear group increases, that is going to decrease the activity of the ventrolateral preoptic nuclei. As a result, what did we see here? So there is no inhibition of the ventral from the inhibitory connection from the ventral preoptic nuclei to the brainstem nuclear group. So now this ventral preoptic nuclei is now kept under inhibition by the brainstem nuclear group. When this is kept under inhibition, will there be inhibition of the orexinogenic neurons? No. So that is going to be lost. So the orexinogenic neurons are not inhibited now, and now the orexinogenic neurons will be very active. It is going to keep the brainstem centers excited. It is going to cause excitation of the dopaminergic neurons as a result of an increased activity happening in the brainstem nuclear group. Now the switch turns in favor of an awake state. So now persons are awake. And if you look at the nuclear groups and the major system responsible for wakefulness, this is the system. That is the locus coeruleus noradrenergic system. Yes, there are other neurotransmitters as well, like serotonin, histamine. We have seen about these neurotransmitters. But of these neurotransmitters, the major system is the locus coeruleus noradrenergic system. Coming to the switch, how it works during sleep. Let's see how this switch abruptly turns in favor of sleep. So we know that when a person moves from an awake state to a sleep state, they naturally enter into the first phase of sleep, or the first stage of sleep, which is the non-REM phase of sleep. How is this happening? So suddenly what is happening? So we are active throughout the day. As a result of activity, what is being utilized? ATP, energy. So ATP breakdown products will be there, will get accumulated, and one ATP breakdown product, adenosine. Yes, do you remember? We learned about adenosine under the classification of inhibitory neurotransmitters responsible for inducing sleep. So adenosine gets accumulated as a result of increased activity. And what is the role of adenosine? It inhibits the brainstem nuclear group through its connections. So adenosine inhibits the brainstem nuclear group in the locus coeruleus especially. So as a result, when this activity of the wake promoting neurons is going to decrease. So when the activity decreases in the brainstem nuclear group, so it is a flip-flop model. When one activity decreases, the other activity takes over. So the activity of the VLPO neurons, that is going to increase. So this is going to cause, VLPO is now going to cause more inhibition of the brainstem nuclear group. And this switch turns in favor of inhibition of the brainstem nuclear group. So this mutually inhibitory connection. So VLPO releases its inhibition, its own inhibition, when the switch turns in favor of an increased firing of VLPO. So this is now disinhibited. So now what is this going to do? This is going to inhibit the orexinogenic neurons. So when orexinogenic neurons are inhibited, yes, these orexinogenic neurons are not going to cause any neuromodulation. It is not going to cause any more neuromodulation in the brainstem nuclear group, in the dopaminergic nuclear group. And as a result, what is going to happen? So there is no input from the brainstem nuclear group to the cerebral cortex. So as a result, because of no input, now the switch turns in favor of sleep. And which phase of sleep? REM, non-REM phase of sleep. So this is how a person moves from an awake state to a sleep state. So there is an abrupt change, especially involving the neurotransmitter, the neuromodulator adenosine. So this is what happens during sleep. So now, and the non-REM sleep. So what about REM sleep, rapid eye movement sleep? So every sleep cycle, you also have a few minutes of REM sleep, which interrupts the non-REM sleep. So what happens, and how is the switch working here? From a non-REM, how is the person moving to the REM sleep? So the same model, when the activity increases in one set of neurons, that is going to suppress the activity of another set of neurons, or when one set of neurons are suppressed, the other set of neurons will take over. So here it is a balance between the monoaminergic system and the cholinergic system. And remember, when we studied about the neural centers, I told you about the neurotransmitter acetylcholine and the centers there in the brainstem in the pons, which are responsible for the REM phase of sleep. So for the REM phase of sleep, those centers have to be activated. So here, these monoaminergic neurons are REM off cells, if you can connect it to what I told you earlier. So the monoaminergic neurons are the REM off cells. As a result of sleep, what happens is there is no input from the noradrenaline system. So noradrenaline system is constantly suppressed and the cholinergic system takes over. And there is activation of the neuronal groups in the tegmental nucleus, in the lateral tegmental nucleus and the pedunculopontine tegmental nucleus. So when there is activation in the cholinergic system, so what is this neuronal group going to do? So this is going to activate another neuronal group which is there in the pons, which is the nucleus reticularis pontis oralis and nucleus reticularis pontis caudalis, situated in the pons. And this nuclear group is also secreting the neurotransmitter acetylcholine, and this is responsible for the induction of REM sleep. So a person moves into REM sleep initiation. and also through its connections. So there are some complex connections involved here. I don't want to deal more about those. I don't want to deal about those connections here. And they are also responsible for the rapid eye movements which happen during the REM phase of sleep. So this is how persons enter into the REM phase of sleep. And one important feature of REM sleep which we saw in the previous slide itself is muscle tone decreases during REM sleep. So how is that working? So not only the neurons in the nucleus reticularis, pons oralis and caudalis, you have another nuclear group which is activated as well. This is a glutaminergic neurons which are present in the sublaterodorsal nucleus in the pons, and they are going to activate an inhibitory interneurons. And where are these inhibitory interneurons, or where are these going to inhibit? Which neurons are these going to inhibit? These are going to inhibit the neurons which are involved in the spinal cord, which are the spinal motor neurons, and the spinal motor neurons are going to be inhibited. So the muscle tone decreases and results in a typical feature of REM sleep which is called REM atonia, which occurs in REM sleep. So this is how the switch happens from a non-REM to a REM phase of sleep. So the monoaminergic system, responsible for the awake state, and the cholinergic system also, not only during awake state, they also play a role in the REM phase of sleep. So this is the flip-flop switch model for awake state and then the sleep state, and in the sleep state we have seen the non-REM switch as well as the REM switch. Yes, so whatever we have learnt only, we are just going to revise some of these in these slides. So the role of adenosine in sleep, we have already seen what is adenosine. It is a breakdown product of ATP, and what is it going to do? So this, as a result of accumulation, it is going to inhibit the brainstem neuronal group involved in keeping us awake. So, and this is, remember, you must have learnt about the two theories of sleep, the homeostatic theory and the circadian theory. So this is the one responsible for the homeostatic pressure to sleep. So as a result of increased accumulation, this is going to inhibit the brainstem centers responsible for the awake state, and thereby they are going to cause disinhibition of the neurons in the BLPO, thereby they are going to promote sleep. So this adenosine can be inhibited by substances like caffeine. So whenever we feel sleepy, when we read, and sometimes we feel very tired and very sleepy. So keep us alert and awake. What do we have? We normally tend to have coffee. So what does this coffee contain? Caffeine. So this caffeine, what does it do? It actually inhibits adenosine. It is an adenosine receptor antagonist. Thereby it keeps us awake and alert. So that is the role of adenosine in sleep. What is the role of melatonin in sleep? So melatonin is a neurohormone produced by the neurons in the pineal gland and released into the bloodstream. And where do they act? They act on neurons in the suprachiasmatic nucleus. We have already seen about this. This is the biological clock. They inhibit the firing of the neurons in the suprachiasmatic nucleus, and by doing this, they disinhibit the sleep center in the ventrolateral preoptic nucleus. And what is the stimulus for the secretion of melatonin? Darkness. So there is something called dim light melatonin onset. If you look at that, so before a few hours of sleep itself, melatonin level will increase. And this is going to inhibit the wake promoting centers, and it is going to stimulate the sleep promoting neurons. What is the role of the suprachiasmatic nucleus and melatonin in sleep? Let's see more about this. So the suprachiasmatic nucleus, when is it activated? So whenever there is a light signal. So there are fibers going through the retina acting on the suprachiasmatic nucleus, that is going to increase the firing of neurons in the suprachiasmatic nucleus. And when the suprachiasmatic nucleus fires, what is going to happen? So the fibers from the suprachiasmatic nucleus, they do not directly innervate the sleep center. What do? Say they go and act on another center in the hypothalamus, which is the dorsomedian hypothalamus. So from the dorsomedian hypothalamus, you have inhibitory connections going through the paraventricular nucleus And this paraventricular nucleus, it is going to inhibit the secretion of melatonin from the pineal gland. You will be learning more about this, the circadian rhythm, and more about this melatonin and suprachiasmatic nucleus in the forthcoming lecture as well. So, when melatonin secretion decreases, so the persons are going to be awake. Now, what happens that from the dorsomedian hypothalamus, you have inhibitory connections going to the ventrolateral preoptic nucleus. So, we have already seen ventrolateral preoptic nucleus is the one responsible for inducing non-REM sleep. So, that is going to be inhibited when the suprachiasmatic nucleus fires. So, melatonin decreased as well as ventrolateral preoptic nucleus is inhibited. So, that is going to result in an awake state. So, what happens during darkness? So, in darkness, there is going to be decreased firing of the suprachiasmatic nucleus. And when suprachiasmatic nucleus firing decreases, this activity in the dorsomedian hypothalamus is going to be less. So, there is going to be less inhibition of melatonin secretion. So, melatonin secretion is going to increase. What will melatonin do? Melatonin will act on the suprachiasmatic nucleus and it will decrease the firing of suprachiasmatic nucleus further. As a result of all these, the ventrolateral preoptic nucleus, it is going to be released from this inhibition. So, there is going to be disinhibition of the ventrolateral preoptic nucleus. So, in darkness, disinhibition of ventrolateral preoptic nucleus, sleep is going to be induced. So, suprachiasmatic nucleus, it is not directly connected to the sleep centers. So, through the other nuclear groups, it is connected to the sleep centers, and that is how it is responsible for the circadian rhythm generation and for the pacemaker activity. Yes, so now we have also seen about melatonin and its role in sleep. We have seen about all the neurotransmitters which are involved in sleep and how are they responsible for the sleep. So, just a summary of the neurotransmitters responsible for arousal and sleep. Yes, so these are all the neurotransmitters responsible for arousal and sleep, and noradrenaline is the most important one responsible for keeping us awake and alert. And you have the other neurotransmitters, serotonin, histamine, dopamine, glutamate, orexin or hypocretin, and acetylcholine. Acetylcholine has a role not only in wakefulness, also during REM sleep. Then sleep promoting neurotransmitters. So, the sleep promoting neurotransmitters are GABA, melatonin and adenosine. GABA, a very important neurotransmitter which is involved in induction of sleep. So, this is the summary of these neurotransmitters, and these neurotransmitters not only work during arousal and sleep state. By doing this, they also have other functions. What are the other functions? So, if you look at serotonin, it is also a mood stabilizer. Acetylcholine, it is implicated in learning and memory. Dopamine, in reward and motivation. So, when this is affected, when there is an imbalance in the secretion of these neurotransmitters, all these can result. There can be learning disabilities, there can be mood disturbances, and there can be disturbances in motivation. All these can occur because these have other functions as well. Yes, so this is the clinical implication of knowing about the neurotransmitters. So, the sleep disorders of late, you can see lot of disorders attributed to lack of sleep or any other sleep disorder. So, it has become a very major, not only a mental health issue, also it is contributing to other diseases, other physical diseases. So, imbalance in neurotransmitters, psychiatric disorders and lot of other disorders are linked to it, and mood disorders, lot of things are now implicated to the imbalance in neurotransmitters. So, if you look at this insomnia, all of us must be familiar with this term already. What do you mean by insomnia? Lack of sleep is insomnia. So, we now know the basis of the neurotransmitters. Now, we can easily say which activity increases, there is going to be lack of sleep. Activity in the wake promoting neurons, if it increases. Which is the major neurotransmitter involved in wake promotion? Yes, we have already seen the locus coeruleus and the norepinephrine group. So, the noradrenaline locus coeruleus activity, if that increases, and there is going to be less activity of GABA, and thereby persons are going to be kept alert and awake, thereby resulting in insomnia or lack of sleep. Similarly, the opposite will happen, that is hypersomnia, if there is going to be overactivity of the GABAergic centers compared to the monoaminergic system. So that is going to result in hypersomnia. And these neurotransmitter receptors are the sleep drug targets. So we know, we have learned about these neurotransmitters, and then if you look at these neurotransmitter receptors, it is being studied a lot, and it is being used a lot too in the clinical cases as drugs for the sleep disorders. So now let's look at the physiological basis of drugs that influence sleep. If you look at the physiological basis of drugs that influence sleep, yes, so either they have to increase the awake state or increase the sleep state. So you have sedative group and then wakefulness promoting drugs. So I'm just going to tell you only the basis. You will be learning more about it in the pharmacology session. So this will help you to understand the drugs involved in or given in sleep disorders. So the sedatives are the ones which will increase the level of the inhibitory neurotransmitters, that is GABA and melatonin. So GABA agonists, melatonin agonists. Melatonin can be given as a drug. Melatonin is given as a drug to induce sleep. And the melatonin receptor agonists. So these are all drugs which can promote sleep. And the other neuronal system, that is the monoaminergic system, if that is antagonized, okay, if that secretion decreases, then sleep can be induced in that case as well. So either it has to be agonist of the sleep-inducing neurotransmitters or antagonist of the monoaminergic, cholinergic and the dopaminergic system. So the H1 histaminergic antagonist, muscarinic antagonist, serotonin antagonist. So all these are the ones which can induce sleep. And coming to the wakefulness promoting drugs, we know the monoaminergic system, right? So the monoaminergic system, if there are stimulants of the monoaminergic neurotransmitters, that is going to promote wakefulness. And then the activity, not only the level of the neurotransmitter increases, sometimes you can increase the activity of the neurotransmitter which works at the synaptic level. So remember, we saw about the transmission, how it happens at the synapse, and how this transmission terminated. Usually by two steps, either of these, the transmission is terminated, either by taking back the neurotransmitter into the presynaptic terminal, which is called reuptake mechanism, or the neurotransmitter can be degraded by enzymes. So by targeting the reuptake, okay, by giving reuptake inhibitors, you can prolong the activity of the neurotransmitter at the synaptic level. And these, especially the serotonergic reuptake inhibitors, these are all drugs which are frequently used in psychiatric mood disturbances disorders. And then coming to the adenosine receptor antagonist, we have already seen caffeine is an adenosine receptor antagonist which can promote wakefulness, and then GABA antagonist. So all these are drugs which can promote wakefulness, and these are all the sedative groups of drugs. So I've told you about the physiological basis. You will be learning more about it in the pharmacology session. So in this lecture, we have done about neurotransmitters, a basic introduction to the neurotransmitters and how the neurotransmitter transmission works. We have seen about the classification of the neurotransmitters. We have seen about the centers where these neurotransmitters are synthesized and released, and how are these connections working in the neural centers. And we have also seen the clinical implications of knowing about it, and we have also seen about the homeostatic pressure to sleep and the role of adenosine, and the circadian rhythm, the role of melatonin we have seen. So this is the summary of what we have seen in this lecture, in this module. These are my references. Thank you, all of you.